Hi students, welcome to UNS classes. We are again back with previous year questions from biology NEET. So let's directly go to the question. Here today the question is complete the flow chart on central dogma. Right? For this we should know what is central dogma. But see the question. Here the question is pDNA. See here it is written that pDNA gives mRNA. Above the arrow there is Q and mRNA gives S and above the arrow there is R. So the question is today we have to find out what P, what Q, what R and what S is. So before going to the answer, as usual, let's go to little explanation about what central dogma is. Okay. So what is central dogma? If we simply say central dogma is nothing but converting the informations present in the DNA to a protein, right? So, the first person who proposed this was Francis Crick. Francis Crick in 1958 proposed a central dogma and he said that the information present in the cell, where the information is present in the cell is the DNA. The information present in the DNA is converted into protein. This process is called central dogma, and the question they asked, what are the different steps involved in central dogma, right? So, what are the basic steps in central dogma? That is replication, transcription and translation. So, if you see this image, see when there is a DNA has to replicate, right? So, DNA normally undergoes replication where the different copies of the same DNA is made, right? After that, this information in the DNA is copied into an RNA, or we can call it as mRNA, messenger RNA, right? From this messenger, so this process is called transcription. When the information in the DNA is transcribed into an mRNA, that process is called transcription. Before transcription, what was the process here? That was replication. After replication, the information present in the DNA is copied into a single stranded RNA or the mRNA is called transcription. After from this mRNA, the cell will translate. The cell will translate the information into proteins, right? And this process is called translation. Or we can say that protein synthesis takes place by reading the information present in the mRNA and the protein is formed. And one more thing is that this process is unidirectional. That is, once this process has been done, we can't go back from protein to RNA, RNA to DNA. Right, so this process is unidirectional. Now let's go back to the question. Here in the question they have written that pDNA gives mRNA, mRNA gives S, right? Here the question is what is S, what is R, what is Q and what is P here. Right. Now we know the steps of central dogma. First is replication. From replication, the information is copied to a single strand mRNA, that is called transcription. Right. Something is transcribed from the DNA to RNA, and from the RNA the protein is made, and that process is called translation. That is, whatever information present in the mRNA is translated into a protein. The process is called protein synthesis. Right. Now let's fill in what will come here. So here P, so DNA normally undergoes replication, right? Replication. Okay, then from DNA to mRNA, what is Q here? What is this process? DNA into mRNA, something is transcribed. The information present in the DNA is transcribed into a single stranded mRNA. So this process is called transcription. Transcription. Now from mRNA something is made here. From mRNA the information is translated into a product called protein. So what will S here? S will be protein, right? And the process is called translation, right? Because the whatever written information in the mRNA is translated into a protein and the process is called translation. Right, it's clear. Now it's very easy to find the answer. If we have this order, replication, transcription, translation and protein, that will be the answer. The first P. P should be? P should be replication. Where is replication? In these two options, P is replication. Okay. Then what is Q? Q is transcription. In which two option we can find? The both option Q is transcription only. Then comes R. R, what is R here? R is translation. In this option, it is transduction, not translation, but in this option it is translation, right? And what is S? S is protein. In this option it's protein, in this option also is protein, but here the step R in this option is transduction, so the correct option will be D. Then what is transduction? Transduction is nothing but injecting the another genetic material into another body. Normally viruses and all do that. That's called transduction. That has nothing to do with central dogma. So if we say central dogma, what is central dogma? Replication, transcription and translation. Right? Hope you got the concept. Happy learning.